Hello friends, today we are going to study about protein folding. How does a protein fold to its native conformation? Let's understand this with an example. Suppose a protein consists of 100 amino acid residues. Each amino acid can take approximately of 10 conformations. In total, this complete protein, how much conformation it can take? That is 10 to the power 100. So many conformations. Now, if, say for example, 10 to the power minus 13 seconds is needed for one conformation, for a protein to check for one conformation, then to finish this much conformation, it will take 10 to the power 77 years. So the process of folding can't be random or trial and error basis because a cell or a biological system cannot spend 10 to the power 77 years to decide a proper conformation for a particular protein. So there must be a shortcut method by which protein folding is taking place inside our cell. It can't just be a trial error or random based method. So that means the protein folding is a directed pathway. It is a well-designed directed pathway that leads to a folding of a protein to its native conformation. So protein takes less than few seconds to fold. Why? Because it follows a directed pathway, which is just not a random pathway. And this folding is a one-way process because the protein folds increase the stability of that particular protein. So that's why the folding of a protein is a proper directed pathway. Now, different models were proposed by which a protein can fold. The first model is a hierarchical model. That means the protein folding will start from assembly of secondary structures. They'll join to form supersecondary structure. More such supersecondary structure will form tertiary structure. It will form a motif, domain, a polypeptide. Similarly, multiple polypeptides will join together to form a complete protein. This is known as hierarchical method. So this hierarchy is followed for the assembly of the protein. Now, the second model is a spontaneous collapse. Spontaneous collapse means the peptides and polypeptides, they will spontaneously will collapse with each other. And when they collapse, the hydrophobic amino acids present between them will start interacting. And that's how they start aggregating with one another, forming a complete protein. So either they are following a hierarchy or they are just spontaneously collapsed with each other, generating a hydrophobic interactions, and that's how they start aggregating. When they collapse with each other, that is known as molten globule. That collapse state is known as molten globule. But nobody knows that exactly which path or which model a protein follows for its folding. It is believed that proteins are not fixed to a particular pathway. They use multiple models for their folding. So they can even use the combination of both the models for their proper folding. Now folding is a cooperative process. Cooperative process means the folding of one state will increase the chances of its further folding. So that is known as cooperative process. Now, as we all know, thermodynamically, the process will be favorable. Why? Because the completely folded protein is having lowest Gibbs free energy change. The Gibbs free energy of unfolded protein is more. Similarly, entropy for unfolded protein is more and the folded protein is less. So thermodynamically, folding of protein is favored. This process of folding protein will be a continuous process till a state is reached when it will have its lowest free energy. And this is expressed by a graph known as folding funnel graph. So what the graph is showing, as the folding of protein progresses, its free energy content is decreasing and the entropy is also decreasing. So thermodynamically, the protein folding process is favored, and while folding, with each step, the energy and the entropy will go on decreasing. So it is given by this graph known as folding funnel graph. Now proteins have evolved to have efficient folding pathways as well as a stable native conformations. So when the process of folding is going on, It is not a single step process. The open polypeptide, it will fold, then it will fold more. So like this, the process will continue. So during this process, there are some stages when the protein can be in their semi-stable form. And then it again proceed to have a stable native conformation. Now for proper folding, or particularly if I say for proper formation of a disulfide bond, an enzyme is needed which is known as protein disulfide isomerase. Now, as the name suggests, the function of this enzyme is to form a proper disulfide bond between the amino acids in a particular protein. Now, how it works? It can work in two ways. This is the first way. What is happening here? This enzyme PDI is already in a reduced state, SH. The protein is already folded, but the bond formation between this cysteine residue is not a proper bond formation. It should be in its native form. Here, the cysteine at position 1 is bonded with 2 and at position 3 is bonded with 4. Now the enzyme will come, which is in a reduced state. This SH will attack this first position of cysteine, attack this cysteine which is at first position, and it will form bond with this particular cysteine. Now the second is free. So what it will do? It will come and attack the third position and form bond with it. When the bond is formed between second and third, the fourth became free. So what it will do? It will go and attack the cysteine of first position, releasing the enzyme. So now the bond formation is between 1 and 4 and 2 and 3, which it was earlier 1, 2 and 3, 4. Here the bond formation was 1 with 2, 3 with 4. Here the bond formation is 1 with 4, 3 with 2. So this is how protein disulfide isomerase works for proper disulfide bond formation within a protein. The second way it works is, in this case, the enzyme is already in an oxidized state. Means it is already bonded with itself. Now, here in the protein, the amino acid cysteine is free. This sulfur can attack this enzyme and it can form bond with this particular cysteine in the protein. Now, when it forms bond with the protein, this SH group is free. Now, this second position will attack the first position and they'll form bond, releasing again the enzyme from it. So by two ways, protein disulfide isomerase can help in the formation of proper disulfide bond. Either it can be in a reduced state or it can be in an oxidized state. Apart from this, one more enzyme is needed, that is peptide prolyl cis trans isomerase. So what it does? We already studied in the peptide video that the amino acid proline, it is not always in a trans configuration in peptide bond formation. There are cases when it is present in a cis form even. So if the protein peptide bond involves proline with cis conformation, during that time this enzyme is needed for its conversion to cis to trans form, so that a proper bond can be formed. So this is how these enzymes play an important role for proper folding of a protein. Now one question for my end for you all: why the folding funnel graph sides are not smooth? Why breaks are present in the graph? Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.